cloudy. When sampling the bacteria or the broth, he noticed that bacteria was present. So the proponents or the believers of spontaneous generation were excited, and they said that this experiment proved the theory of spontaneous generation. Not all the scientific community was convinced, so we had to redo this experiment. Next came the experiment by Spallanzani. Spallanzani, who was another clergyman, but this time of Italian descent, repeated that of John Needham's experiment. In this case, he again did a container containing broth, allowed to let it cool at room temperature, and whether he sealed the container or not, both did appear. So Spallanzani actually brought this one step further. In this case, again, heated the broth to destroy the living organisms, the microorganisms. This time, he immediately sealed the flask. Small change in the experimental design led to the finding that microorganisms would not grow, were not detected in the broth. Only when the flask was opened up, allowed later to sit at room temperature, did the microbes enter the broth. I have a question for you. What happens when you actually boil the container and seal it before you allow it to cool, before you allow the contents to cool at room temperature? You're creating is actually a vacuum seal inside that container. But the believers of spontaneous generation didn't want to let this go, so they quickly said that, well, there's some vital component that must be present in this vessel in order to allow the microorganisms to spontaneously be derived from the broth. The only thing that's absent in a vacuumed container is that of air. There was no air flask. These proponents or these believers of the theory of spontaneous generation said that air is a vital component. Means that air needs to be present in order for organisms to be derived spontaneously from non-living matter. So, again, this experiment didn't lead to anywhere. We had to wait for one more experiment. That, I have to bestow the honors on Louis Pasteur, French scientist in 1859, who designed an experiment that was prove or disprove spontaneous generation once and for all. Give you a little bit of background about Pasteur. He strongly believed that microbes are present in the air. He also believed that microbes were the cause of making people sick. If microbes were present in the air, he believed that these microbes could be spread from one person to another, which is the basis of the germ theory. Pasteur introduced the idea of the germ theory, but did not prove it. We're going to have to wait for another person. Before we go on, let's describe the germ theory. Theory states that some diseases caused by microorganisms. Diseases that are caused by microbes, to distinguish them from other diseases, we're going to call them infectious diseases. So let's go back to the experiment that Louis Pasteur proposed and performed in order to prove or disprove spontaneous generation once and for all. Again, Pasteur, just like Needham and Spallanzani, started working with a flask that contained broth that initially had bacteria or microbes present in it. Like the other two scientists, he boiled the broth, killed all the organisms that were present in the broth. What he did differently is he had a flask that he curved at the neck. We call this the swan neck flask. Remember that Pasteur believed that microbes were present in the air. If that's the case, air could enter the swan neck flask, but it would get trapped in the bottom angle here. Air would not work its way out up. Well, actually, the air could, but the microbes that were present would stay stuck in the neck of the flask. The air could enter and exit the flask, so he's not creating a vacuum seal. So the vital component, air, after the flask was allowed to boil, it was cooled at room temperature. Again, the only difference is that the neck of the flask, it was left open. There was no sealing, but it was at an angle. The flask remained incubated at room temperature for weeks, for months, and at no given time did we see any microorganisms present. Again, simple adjustment of the neck, this curvature of the neck, allowed Pasteur to finally disprove the theory of spontaneous generation. Again, some people could say, well, the broth wasn't able to support life if the organisms were going to grow spontaneously. So he was ahead of the game. The neck, this flask here, this allowed the air to enter easily with any dust particles that might be present in the air. Microbes were able to enter the flask and start growing. And microorganisms were present. This showed that the broth, given the chance, could support the growth of microorganisms. Clearly today we don't believe in the theory of spontaneous generation. We believe that living organisms are derived only from other living organisms. So the theory of biogenesis was the point of going through this history of these experiments trying to prove or disprove spontaneous generation. The whole point is to show you the power of well-controlled or well-designed experiment has the proper controls. In this case, Pasteur did not seal the flask, so he did not create a vacuum. He left the vital components of air present in the flask. He allowed so that only the air would enter the flask and not that of the dust particles and the microbes. So a well-designed experiment goes a long ways. Since then, the invention of the microscope and knowing that microorganisms are derived from microorganisms, come to discover many types of microorganisms. And we can divide them into these 7 types listed here. I told you that microorganisms are the study of organisms that are too small to be seen by the unaided eye. Reason being is that I want to make a point to say that viruses are the only non-living microorganisms. Discuss this more in my video on viruses. We can also divide these other types of microorganisms. We can say that the bacteria, the archaea, are eukaryotes. These are organisms that are made out of cells that do not have a true nucleus. We're also going to say that these are the most diverse. Discuss that in another video. The other microbes, the fungi, the protozoa, the algae, and the helminths, be single cell, could be multi-cell. They are all eukaryotes. Eukaryotes. That means that all their cells contain a nucleus. We learned more and more about these 7 types of microorganisms. Discovered that most of them live as a free existence. That means that they don't rely on other organisms. They can live independently in an environment that's rich enough to support their growth. For the most part, most of them are also harmless and actually are needed for our growth. So they're what we call beneficial. They help us. However, for a small portion of these organisms, they actually have to live closely associated with other living organisms. These organisms that they are needed for their growth are called hosts, and the organisms are called parasites. It's these guys that give microbes a bent. The ones that cause disease. The germ theory. Along with the study of microbiology, we've identified quite a few microorganisms. Just like the study of any other biological forms, we had to categorize these organisms and give them a name. underlined or italicized is going to be the species name. Sometimes you're going to see it referred to as a specific epitaph.